Today we have Professor Marina Sirota joining us to talk to us about her work in Alzheimer's disease. Marina is currently a professor and the interim director of the Bakar Computational Health Sciences Institute at UCSF. Prior to that, she has worked as a senior research scientist at Pfizer, where she focused on developing precision medicine strategies in drug discovery. She completed her PhD in biomedical informatics at Stanford under the mentorship of Dr. Atul Butt. Dr. Sirota's research experience in translational bioinformatics spans nearly 20 years, during which she has co-authored over 170 scientific publications. Her research interests lie in developing computational integrative methods and applying these approaches in the context of disease diagnostics and therapeutics with a special focus on women's health. The Sirota Laboratory is funded by NIA, NLM, NIAMS, Pfizer, March of Dimes, and the Burroughs Wellcome Fund. As a young leader in the field, she has been awarded the AMIA Young Investigator Award in 2017. She leads the UCSF March of Dimes Prematurity Research Center at UCSF, as well as co-directs ENACT, a center to study precision medicine for endometriosis. Dr. Sirota is also the founding director of the AI for All program at UCSF, with the goal of introducing high school girls to applications of AI and machine learning in biomedicine. And with that, I'll hand it over to Dr. Sirota.
Oh my gosh, what an introduction. Thank you so much. Thank you for having me. And it's my pleasure to be here and share some of our work bringing different types of data together to hopefully enable predictive medicine. And again, I'll be using Alzheimer's disease as an example, but we work on a variety of different disease areas in the lab. And as I understand, there's a lot of trainees in this in this venue. Is that correct? Is
Yeah,
this
that's correct. Yeah.
okay? So please make it as interactive as we can via Zoom. I'm sorry I can't be there in person. Please interrupt me and ask questions. It's really meant to be a conversation. So I always like to start by saying why I'm excited to be a computational biologist. And the reason is there's a lot of data out there, and we have to try and figure out how to make sense of that data. There are many publicly available data sets, which include transcriptomics data from the Gene Expression Omnibus. The cancer community has developed the Cancer Genome Atlas. There are clinical trial data sets in research repositories like Import, and there are many, many, many others. Also, as the technologies advance, we are coming up with more creative ways to explore omics. Genomics and transcriptomics have been around for a while, but we can also look at the antibody repertoire, epigenomics, proteomics, metabolomics, the microbiome, and now many, many of these on a single cell level. And finally, the third type of data that I'm really excited about is clinical data. This is real world information that gets captured when somebody goes to see a physician, and it includes diagnosis codes, lab test results. We have de-identified clinical notes that are available for mining. And really, it's quite amazing. And we're very, very fortunate at UCSF. We have access to this data for UCSF itself and across the UC system. And these are all efforts that were spearheaded here at UCSF by our inaugural director, the late Dr. Atul Butt. So to give you an idea of what we have access to, we have information, the slide is a little bit out of date. It's 9 million patients, and that's the structured clinical data, over 135 million encounters and 40 years of activity. We have de-identified clinical notes that can be mined. We have imaging information, over 82 million images. And then we have some genomics, though I'll be honest, the genetics and genomics data that we have on the UCSF patients is quite limited. It's limited to a cancer genome panel called the UCSF 500, and we have that on about 20,000 cancer patients. All the data is de-identified, it's accessible and computable. So anybody here can take advantage of it. You have to do a little bit of training, but anybody can have access to the data and start mining it, which is quite amazing. We also have data across the UC system. So you can think of the UCSF data as deep and the data across the UC system as broad. This is 6 medical centers. UC Riverside and UC Irvine are pretty small, but nonetheless, UCLA, UC San Diego, and UC Davis are considerable. It's 12 years of data, so it's since the EPIC implementation across these entities. It's also about 9 millions of patients, and we have data on about 40,000 cancer genomes. And you can appreciate that this is truly an opportunity for a discovery and validation system where we can discover on the deep data at UCSF and then validate across the UC system. And these efforts were also spearheaded by Dr. Butte. We have opportunities to explore these data with large language models. Early on, we have, and we still have, access to Versa, which is an HIPAA-compliant implementation of being able to interact with both the unstructured data as well as the clinical notes with a variety of different large language models. And UCSF, and these are all efforts led by Kilai in the our larger IT group. And now we just announced a few months ago, the chancellor announced a partnership with OpenAI, so we'll be able to partner with them on some of these analyses also. So I'll pause for a second, but during this presentation, I'd like to tell you two stories. One more in the therapeutic space, looking at drug discovery and leveraging both molecular and clinical data for drug discovery, and the second one more in the diagnostic and disease prediction space. But before I jump into the science, any questions? Okay, don't be shy though. All right, so in my lab, we try and figure out how to put different types of data together, both molecular data, mostly from the public domain. We start usually with that, but then we partner with collaborators to generate data sets that we don't have or data sets that we'd like to have. And then also using clinical data, the resources I just mentioned. A lot of the work in my lab focuses around women's health, reproductive health, like pregnancy outcomes, preterm birth, and recurrent pregnancy loss. As Joe Wong alluded to, we study endometriosis pretty extensively. We have a large research program in autoimmune disease. As many of you probably know, autoimmune disease is a lot more common in women than men. So we try to understand why and what might be the molecular underpinnings of that. And then we study neurodegeneration and Alzheimer's. So you can see kind of we go across the life course. Due to time constraints, I'm going to focus, and also interests of this group, as I understand, I'm going to focus on Alzheimer's disease as an example, but we do similar type of work across, as I mentioned, across the life course. So drug repurposing, or the idea that we can use FDA-approved drugs and try to identify new indications for them is a very, I guess, it's an enticing concept. It is very expensive and it takes a long time to take a drug and bring it to development. Often takes over 15 years and millions and hundreds of millions of dollars to develop a drug and bring it to market. And 90% of drugs fail in that early development time. So a solution to that could be drug repurposing, which again is identifying new uses for drugs that are already FDA approved for something else. It is much cheaper to do that because a lot of the safety testing has been done already, and it also reduces the risk of failure. This idea of computational drug repurposing actually stems from my graduate work at Stanford with Dr. Butte, where we wanted to take a holistic approach. There's a number of different approaches you could try for drug repurposing. You could look at genetics and look at specific targets. So are there certain genetic targets that are shared across diseases, and can then you repurpose a drug based on their known drug target, based on genetics for a given disease? And that has been done extensively. You could look at what we call disease-based drug repurposing. Let's say two diseases appear similarly. They maybe have similar symptoms. They look similarly based on their clinical or molecular data. And one of those diseases has a drug that works. You could try it for the other disease. And that has been done extensively for autoimmune diseases, actually. There are molecular modeling approaches and docking-based approaches that look at the interaction of a drug and its target, and that it can be modeled computationally. I'm not an expert in those approaches at all, but there are many people, both in industry and academia, working in that space. And finally, there's this idea of a network or global signature approach, where the thought is one target, one drug, one disease won't work well for a complex disease like Alzheimer's or endometriosis. And this is the approach that we've taken. So in the very early days, again, this is in my graduate school days, we took all the disease data out of the gene expression omnibus. I imagine these stats are not entirely up to date either, but it's extensive. At the time of this, of when I created the slide, which maybe is a few years ago, there were 8 million samples in Geo, but the more impressive number is 264,000. That's 264,000 experiments, each one, well, many of them dealing with disease. And on the drug side, at that time, we used something called the connectivity map. It was developed by the Broad Institute, and they've taken human cells, they put drugs on them and measured expression before and after treatment. The initial data set had 164 drugs. The expanded data set had over 1,000 drugs. Then there was another effort called Links that you might have heard about, where it's not genome-wide expression, but they took about 1,000 landmark genes with the idea that the rest of the transcriptome can be imputed from that 1,000 genes. And they tested tens of thousands of compounds using that kind of limited transcriptomic signal. In our hands, that data has been too sparse and the imputation hasn't been good enough to pursue the type of work that we want to pursue. So we focused on this middle layer. And I'll allude to it at the end, but more recently, there's been much more extensive data sets that came out, even on a single cell level, such as the Tahoe 100 million data set that captures expression before and after treatment with a drug on a single cell level across thousands of cells. But the initial study, well, the initial study was done in 164, but the majority of the work that I'll talk about today leveraged this middle, kind of middle bullet point. And it contains both FDA-approved drugs and some non-drug bioactive small molecules. These are all amazing resources. If you want to try and play around with, I highly recommend it. Okay, so again, this is stemming from my graduate work. We developed a computational pipeline for drug repurposing, and it's very, very simple. The hypothesis is, well, for given a disease signature, for every drug, we compare that disease signature to the profile of that drug and ask how likely is that drug to reverse that gene expression signature back to normal. We use a non-parametric rank-based method called the Kolmogorov-Smirnov test. And the hypothesis is if the compound reverses that signature, then it could be a potential therapeutic. So we look for these negative scores. You can think of it as a correlation. We look for these negative reversal scores. And again, you can appreciate it's a very, very simple hypothesis. But the paper, the original methods paper, came out now almost 15 years ago. We've applied it to a number of different disease areas, starting out with IBD and cancer. We applied it to dermatomyositis. Cancer is a natural application of this method because the drugs are tested in cancer cell lines, so that we can really match the expression reversal if we look at specific cancer types. And then we looked at, because I care quite a bit about pregnancy outcomes, we looked at preterm birth during COVID. Of course, we tried this for COVID-19. But today, again, we're going to talk about the applications to Alzheimer's. This is a review paper that we published about 5 years ago. Check it out if you're interested about how computational discovery and drug repurposing has developed for Alzheimer's. So the first application of this approach to Alzheimer's disease came out in Nature Aging in 2021. And this is work of Ali Taubes, who was my first graduate student at UCSF. And this was in a very close collaboration with Dr. Yidong Huang. So she was a joint student between our groups. And the reason we decided to focus on Alzheimer's, it's a devastating disease. And really, the majority of clinical trials have failed to produce a drug that works. You can see that it's complex. There are many different cell types that are involved in different processes from neuronal degeneration. There are amyloid plaques, tau tangles. So it's very complicated. And there are many factors that affect our risk. So it's heterogeneous and multifactorial, including environmental factors. Obviously, aging and genetics are two of the biggest ones, but things like diet, exercise, gut microbiome, oral health, other toxins, all might play a role in predisposing somebody to Alzheimer's disease. Because it's so complicated, it's difficult to find a drug. There have been 68 different trials, and you can see how they've advanced from phase 1 and phase 2 to phases 3 and 4. They target different pathologies of Alzheimer's, but again, there has been very limited success. So that really brought us to think that one target, one disease, one drug, one target, one disease, especially for Alzheimer's, won't really work. So this initial study that was led by Ali, we gathered publicly available data. At that time, it was mostly microarray and RNA-seq data that was pretty old. And we wanted to see if we can apply precision medicine strategy to Alzheimer's. We know that ApoE is the biggest risk factor for Alzheimer's disease. So we wanted to stratify the patients by their ApoE status. So ApoE44 is the most aggressive genotype for Alzheimer's. And the biggest challenge, if you want to study ApoE44 disease, is finding controls. There's very few of them because the majority of patients with ApoE44 genotype get the disease. So this is the data that we worked with, not a huge sample size, but we needed a data set that had both genotyping as well as transcriptomic data. So here you can see each dot is a patient. They're colored by case control. And here's the same visualization colored by genotype. And you can see that the ApoE44 genotype, the red dots here, are mostly on the left-hand side. So there's quite a bit of separation. And of course, many of them are the ones that are cases, but there are some that are controls. When we started looking at transcriptional signatures, we saw that both on the gene level, which is these first two diagrams, as well as the pathway level, there are a lot of effects that are genotype specific. So there are some genes that are uncommon. These are the upregulated genes, that are the downregulated genes. But a lot of the signatures are genotype specific. Again, focusing on the ApoE44 signature, which is here in red. And then we observed the same thing on the pathway level as well. Actually, a lot less similarity on the pathway level, more signals that are genotype specific. And here we're looking at ApoE33 in blue, 4,4 in red, and 3,4 in gray. So then we took these three signatures and we compared them across the drug data in the same way that I mentioned before. And we saw when we focused on ApoE44 disease, there was a drug called bumetanide that was at the very bottom of the list. So strongest reversal, one of the strongest reversals. It was still significant across the other genotypes or in all the samples when all the disease samples were taken together here on the right, bottom right side, but it was strongest in ApoE44 disease. And this is just the visualization of that reversal. So that really got us thinking, and we wanted to explore it further. Bumetanide turned out to be a loop diuretic that works by decreasing the reabsorption of sodium by the kidneys, and it's used to treat edema and hypertension. And we wanted to test it in an animal model, and this was all work done in Dr. Huang's lab. So, I'm not a mouse expert at all, but I'm going to give this my best shot. As I understand, the mice do not like to be in the water. So when they're placed in this pool of water, they try to get out. There is a hidden platform in the target quadrant that allows them to escape. And what we measure here is the time that it takes them to get out and whether they remember that. So this is looking at learning deficit in the animals. So in the black, you see animals that are 3-3, ApoE3-3, so they don't have a learning deficit phenotype. Whether they're treated with bumetanide or not, they do fine. In the red solid line are the animals with ApoE44 gene knocked in. And you can see it takes them a longer time to get out, to escape, unless they're treated with bumetanide. And that's the red dotted line. You can see that it's significantly brought back to more of a normal state. So this was really exciting to us. We followed up with some sequencing, analysis, single-cell RNA-seq of the brains of the animals treated with bumetanide. So this on the right-hand side, you can see each dot is a cell. They're clustered into relevant brain cell types. And then you can see the animals treated with bumetanide in red versus in blue versus vehicle in blue, and there's a significant shift. When we look at that shift reverses that disease gene expression signature that we started with, at least for some cell types, not all of them, but at least for some. And this is in a different mouse model. So we tried two different mouse models here, which was also, it was interesting to us. We also did some electrophysiology. I don't have time to get into that. I want to focus on the computational aspect of things. But the next question we wanted to ask, well, okay, so now we have the human transcriptomic data. We have the mouse data, which seems to support our hypothesis. Can we use real-world electronic medical records and query whether patients on bumetanide have a lower prevalence of Alzheimer's disease? So we went back to the data that I mentioned before and started with the study at UCSF. So at that time, this was a number of years ago, we had access to 1.3 million patients. About 5,500 of them have been exposed to bumetanide. We wanted individuals with demographics and wanted individuals over the age of 65 because, again, we don't want to compare 65-year-olds to 18-year-olds when we're talking about Alzheimer's onset. Once we identified our exposed group, we used propensity score matching with demographics, as well as the indication for bumetanide, which is hypertension and edema. And we carried out 10 times propensity score matching, identifying a control group to compare to. And then we compared Alzheimer's versus not. And this was all work done here at UCSF by Edith Kosti and Tomiko Skotsky. And this is what the data looked like. We saw that across the 10 groups that we looked at, the prevalence of Alzheimer's was lower in the bumetanide exposed groups than the matched controls. At this point, we didn't yet have the data across the UC system. So we partnered with a group at Mount Sinai. This was Benjamin Glicksberg and Masoud Bijak to ask the same question. They had slightly more patients, about 3.9 million. Not everybody had a medication record, so we narrowed down to those that did, about 3,000 exposed to bumetanide, about 2,300 over the age of 65, carried out the same type of propensity score analysis, and saw that the strength of association was even stronger. So again, patients exposed to bumetanide, this bumetanide cohort on the left-hand side, has a significantly lower prevalence of Alzheimer's than the matched controls. So this really gave us an idea that we can use transcriptomics data to come up with a hypothesis, validate it in an animal model, and then further validate it using clinical data. Next, we were interested in exploring combination therapy. Maybe I'll pause there for a second. Any questions? Okay, no questions. All right, so we wanted to see if we could identify combination therapy for Alzheimer's. And this was work led by another amazing graduate student in my lab, Yachao Li. She just completed her training about a year ago and now is a research scientist at the Gladstone Institute with Dr. Huang. This was also in collaboration with Dr. Yidong Huang. So we talked about the heterogeneity of Alzheimer's, and there are many different cell types involved. This is at the time when single cell data started being available. And we really wanted to leverage that to identify combination therapies that might impact different cells. The approach was very similar. At that time, there were three single cell studies that were publicly available. Pretty limited data. Now there's much larger ones, and we're leveraging those. But at that time, we had data on 37 Alzheimer's patients and 38 controls across these two studies, about 180,000 cells. First, we have to normalize them. I'm not going to get into the details. I think this demonstrated the normalization. Before normalization, all the studies clustered separately. but we were able to combine and bring them to the same level, which is shown here. This is colored by case control. This is colored by cell type. And we focused on six major neuronal cell types: astrocytes, two types of neuronal cells, microglia, oligodendrocytes, and OPCs. So unfortunately, we couldn't stratify these further by genotype. Because we had so few samples, we did not have any ApoE44 controls in this set. So we decided to look at the cell type heterogeneity, and we had to bring all the genotypes together. This is what the data look like. Again, before normalization, then after harmonization, we can get all of this on kind of one level, and then colored by cell type. These are the signatures, the up and down, significantly up and down dysregulated genes across every cell type. We can look at the specific genes as well as the pathways, but we really wanted to query them against the drug data. So again, we're leveraging the connectivity map data set, about 1,300 drugs, and we're using now cell type specific signatures to explore the reversal. So for every cell type, we query that data against the drug data to see which drugs might reverse the disease effect in that given cell type, with again the same hypothesis of reversal. So this is what the data looks like. In pink, and these kind of rounded rectangles, you see the various cell types. In the ovals, you see the drugs. We saw that some drugs, for instance, trichostatin A, affected multiple cell types, and we really wanted to focus on those here. The blue ones reverse one cell type, purple and kind of gold colored are 2 and 3 cell types, respectively. And again, we wanted to focus on ones that affect multiple cell types. We want to go back to the EMR. And here, instead of using that at the very end, we wanted to use the electronic medical record analysis to screen the drugs and help us choose the right combination to focus on. So the analysis is very similar. We have an exposed group to the drug, an unexposed group, and we look at the relative risk of Alzheimer's using propensity matched controls. This is the summary of the data. So we tested 25 drugs using electronic medical records, and we got 5 that had significant effects, significantly lower relative risk score on the drug versus not. There were 2 of the top ones, letrozole and irinotecan, that really caught our eye. So here you can see the reversal across the cell types. And you can see that letrozole reversed the signature in neurons, and irinotecan reversed the signature in glial cells. That's how we ended up picking these 2 drugs. They both happen to be cancer therapeutics. So the pipeline started with about 1,300 drugs. 85 reversed the signatures, any signature significantly. 25 were queried. Well, 25 were multi-cell type candidates, and 5 of them held up using electronic medical records. So again, we chose these 2 because they were interesting and also because they were at the top of our list in terms of the EHR analysis, and they affected different cell types. So letrozole is an aromatase inhibitor that blocks the conversion of androgens into estrogens. And irinotecan is a topoimerase 1 inhibitor that interferes with DNA replication. So their mechanism seems to be different. And then Yechao started an animal study where she crossed 2 animal models for Alzheimer's. Again, this is not my area of expertise. She looked at the combination therapy as well as each therapeutic alone, as well as animals who were untreated, and carried out the same water maze test that I told you about earlier. So here you can see that, again, we look at that latency response, and you can see that in the combination therapy, whether we look at the 24-hour probe or the 72-hour probe, that's the latency, how long test the memory in that span of time, there's a significant decrease when treated with the combination versus the individual drugs alone or untreated. So then she looked at a number of other pathological measures. We looked at hippocampal volume and saw that there was a significant increase with the combination treatment. Looked at amyloid beta staining, p-tau staining, looked at microglia as well as astrocyte staining, as well as neuronal staining. And this is a summary of all of those results. So each treatment alone sometimes was significant, but sometimes not. However, for the combination therapy, the majority of the factors were significant in demonstrating better brain health. So finally, as the last step of this analysis, and you can appreciate this, so all this work was done by Ye Chao, by a single graduate student, in both, well, she was in my lab, but also she was a mouse biologist, so she did all the mouse experiments. We looked at, again, reversal in the relevant cell types before and after treatment. So this is from a single cell experiment, looking at untreated animals versus animals treated with the combination. And we saw that cell type abundance, there were more CA1 and CA3 cells after treatment. And then we looked at specific pathways, as well as pathway reversal. So here, the AD signature for every one of these columns is on the left-hand side. And you can see that the colors are flipped between that as well as the combination therapy treatment across all cell types that we looked at. And this is just a summary demonstrating that. So here we extended the approach to single cell data on the drug side, on the disease side. We queried cell type specific signatures to identify combination therapy, used EHR analysis to further narrow down the list to the combination that we want to test, and then explored that in extensive animal studies, as well as looking at reversal in the animal model after treatment. So our thinking is that by layering all of this different type of information, we can hopefully address heterogeneity of Alzheimer's a little bit better. And truly, this type of work is only possible in a collaborative setting. Without my collaborators on the wet lab side, there's no way we'd be able to do this type of analysis and work, taking a hypothesis and truly validating it. So again, throughout our Alzheimer's drug discovery work, we started with bulk and then extended to single cell gene expression data to look at combination therapy for Alzheimer's, validated in EHRs, as well as extensive animal models. I believe bumetanide has been moved into a clinical trial. We're hoping to extend that to the cancer drugs as well. And we're expanding this approach to other complex diseases such as endometriosis. We're also starting to incorporate single cell data on the drug side. I mentioned the Tahoe 100 million data set. It's pretty incredible and amazing, and we are in the process of evaluating it against some benchmarks to see what additional information it can bring, and then are planning to incorporate that into our drug discovery pipeline. So next, I want to tell you a little bit more of what we're doing in the electronic medical record space in Alzheimer's, but I will pause there for questions. Any questions?
I have a question, but you might get into it, so maybe I should hold it. But
No, go for it.
I was curious about the phenotyping for Alzheimer in real world data because there's, in conversations with neurologists in the past, I heard that sometimes it could be coded pretty poorly, and then there can be some conflicting information between like whether this person has Alzheimer's, cognitive impairment, or just dementia. So I was wondering how you guys dealt with that in your real world studies.
Yeah, so I will say we are dealing with it now, and I will talk about it in the second section. For the EHR work to query the drug data, we just used ICD codes. So we did a very vanilla analysis. Also, I mean, there's many there's many things to consider, like the timing, how long ago they were on bumetanide or those cancer drugs. Uh, when does the diagnosis of Alzheimer's come in, that like longitudinal aspect of things, which we also modeled very, very crudely in those early studies. So you're bringing up the right question. Hopefully, I'll be able to address some of it in this next section. But yes, these are all things to think about, and it's not perfect, but nonetheless, it's amazing and very rich data. And you have to figure out that balance of what you and your collaborators are comfortable with. And it has to be this back and forth all the time conversation about how you can, how you can trust what you're looking at and how you can break it. Very good question.
Perfect. I look forward to the rest of the presentation.
Sure. Other questions?
I'm just curious. You said, I think that bumetanide, was that a blood pressure medication?
It's a diuretic
Oh,
medication
okay.
for hypertension and edema.
Got it.
So yes, it does lower blood pressure.
Okay.
But I don't know in terms of the mechanism, like whether it has anything to do with vascular dementia and affecting that. That's a lot more experimental work needs to be done to figure out the mechanism of action of these drugs. I also have no idea what the mechanism of action of the cancer drugs is.
Got it. Yes. Okay. Yep. I was just curious. Yep. If like a drug that, yes, had a similar role, if you saw similar results in terms of protection, but yep, it sounds like you're
We
not sure.
didn't do a comprehensive study of other diuretics. Obviously, we could in the EHR. I mean, mouse studies would be way too expensive to do. It's feasible to take a handful of diuretics and see whether that signal would hold up for others. We haven't done that. I believe there's another paper out there where somebody did that a bit more extensively. Bumetanide definitely held up. I'm not sure about the others, though. That's a good
Got it.
question.
Yeah. Thank you.
Sure. Other questions? Okay, I will move on. So now that, our first foray into EHRs was this retrospective drug analysis, but I was very fortunate to have another amazing MD PhD student in my lab, Alice, who graduated her PhD about a year ago and is finishing med school as we speak. And she wanted to explore Alzheimer's and Alzheimer's heterogeneity using electronic medical records more broadly. So this work came out in Nature Communications in 2022. And we identified, in this first study, identified an Alzheimer's cohort using ICD codes, not ideal, but it was a starting point. Looked at patients over the age of 64 and then used propensity score matching, very similar to the strategy we applied for the drug data, but now looking at disease. So these are patients who did not have a diagnosis of Alzheimer's, but had the same distribution of race, age, sex, and we also matched them by whether they were alive or not. We looked at demographics, diagnosis codes, lab tests, and as well as medications. And analyze those in different ways with also a focus on sex differences. Because as I mentioned, Alzheimer's is a lot more common in women. We wanted to understand that a little bit better. At the time of the study, or at least at the time that the study was launched in 2020, we did not yet have the UC-wide data. So we partnered with Mount Sinai, similar to the first study on a drug discovery for validation. So each dot is a patient, and it's a dimensionality reduction plot, but we're looking at their clinical profiles. So they're colored by case control, and this is excluding diagnosis of Alzheimer's. And you can see that there is some separation between cases and controls, but it's not like crystal clear, but there's some signal. The cases are in red and the controls are in blue. And you can see that that's the case both at UCSF and Mount Sinai. Then we could build these comorbidity networks. On the left-hand side is the comorbidity network for Alzheimer's versus controls. You can see a lot more connectivity in the Alzheimer's patients. It's not too surprising. It's a more coherent group of patients. And the connectivity that you see is between Alzheimer's and dementia, essential hypertension, disorders of lipoprotein metabolism, things like that. Then we could look at that stratified by men and women. Again, more dense, actually a more dense network in the females than males, some differences, but both are more dense than the controls that are shown here in the middle. We could look at specific associations. Of course, mild cognitive impairment and dementia are associated with Alzheimer's, not too surprising. But as we go down the list, we see things like UTI, hypertension, hyperlipidemia. And in terms of the negative relationships, we saw cancer, and that has been shown before as well. All of these are consistent, at least somewhat consistent, between UCSF on the x-axis and Mount Sinai on the y-axis, about 0.8 correlation across the two, which is pretty good given these are entirely different medical systems with different patients, with different doctors, with everything. We were pretty happy with the concordance. We can look at sex differences, as I mentioned. Some associations, like association with dementia, are significant in both. Others have a greater association in women. Those include UTI, interestingly, osteoporosis, I'll get back to that, hypertension, and depression. Some are more significant in males, like sleep apnea, Parkinsonism, irritability, and some just appear in females. Again, both of these are fairly consistent when you look at comparing UCSF and Mount Sinai, and we stratify by sex. Of course, you see things on this like x-axis here and y-axis here, and there are just some that don't really are some codes that are not captured in one institution versus the other. Looking at the drugs, we see a number of compounds that are used more in Alzheimer's. Those include some of the medications used for Alzheimer's disease. Again, not too surprising. Going back to one of the questions, we did see furosemide, which is another diuretic, show up on the protective side, which was interesting. So there are more opportunities to explore here. And then these are just showing the concordance, well, sex-specific analysis and also concordance between UCSF and Mount Sinai. We looked at lab test results. Again, some lab test results are similar in men and women. Others are sex-specific when we look at association with Alzheimer's disease. All of this is interesting to explore. But what we were interested in doing next is building a predictive model. And this is work that came out in Nature Aging in 2024. And here, going back to that phenotyping question, we really wanted to be 100% certain in the phenotypes of these patients. So we partnered with the Memory and Aging Center at UCSF. And this is a group that sees these patients and phenotypes them regularly, both for research as well as clinically. We mapped our de-identified electronic medical records to the Memory and Aging Center database so that we were much more certain which patients have Alzheimer's disease. And then Alice built models, predictive models, 1 day, 1 year, 3 years, 5 years, and 7 years prior to diagnosis. We validated some of these associations, the predictive features across the UC system, and then we used the knowledge network to understand the biology behind them. So this is what the cohort looked like. We started with 5.5 million patients. When we match to those seen at the Memory and Aging Center and have Alzheimer's, we only get about 3,000. So it's a 1/3 reduction from the 9,000 that we looked at the broader phenotype to about 3,000 that we were comfortable with. And then we wanted patients that had longitudinal records up to 7 years to build those models. And that reduces the data set even further to about 750 patients. And we have a large number of controls. We do matching for the controls because when you build a model, again, you don't want to be comparing an 80-year-old who walks through the door with an 18-year-old when we're looking at Alzheimer's. So we match them based on demographics. And we saw that the models performed okay. 7 years prior to diagnosis, we were getting about 0.7 accuracy on average, which is not amazing, but it's not bad either. We wanted to then look at which features are associated or predictive of Alzheimer's. And again, we look across time. So you can see 1 day, 1 year, 3 years, 5 years, and 7 years. These are the broad phenotype categories on the left, as well as the specific ones on the right. And we saw things like memory loss, vitamin D deficiency, osteoporosis showing up across the board. We can also look at this in a sex-specific manner. We can build these models for men and women separately. And we saw that some features were consistent, like hyperlipidemia. Others were specific to women, such as osteoporosis, and others, some were specific to men. And going back to that phenotyping question, you see a lot of the things that we saw from the crude phenotyping analysis as associations in our first study. So while this is much better phenotyping, I don't think it undermines the results that we saw before. The themes are quite similar. So what we wanted to do then is try to understand and dig into these features. So we partnered with a collaborator. His name is Sergio Baranzini. He's a neurologist who developed this resource, a biomedical knowledge graph called SPOKE. And what it contains is existing databases and knowledge bases, about 50 data sets, about 50 million nodes across different types of nodes, 100 million connections, linking genes, drugs, diseases, and so forth. So we could layer that on top of the 25 top features and try to understand what are the biological links between these. So let me zoom into this group that is consistent across time. So when we look at the link between Alzheimer's on the left-hand side and hyperlipidemia, we see that ApoE is showing up. This is not surprising. We know ApoE is the biggest genetic risk factor for Alzheimer's, and it's also involved in lipid metabolism. However, when we looked at the link between osteoporosis and Alzheimer's, we saw a lot of immune genes, TNF, IL-1B, IL-6. This is interesting. It tells us that there may be something going on with the immune system there. So then we wanted to validate these associations. When we looked at hyperlipidemia across the state of California, we saw that the result was significant. And also when we stratify by men and women, it held up. Maybe a slightly less strong association in males, but nonetheless, it was significant across both sexes. However, when we looked at osteoporosis, we saw that this relationship was only significant in females and not in males. So we wanted to dig into that a little bit more. We partnered with Sergio and his team to carry out a co-localization analysis, where we took publicly available GWAS data and one study for Alzheimer's disease and one study for heel bone mineral density. We stratified into males and females, and we plotted the association p-values against each other. And we saw that there was a peak around this gene MS4A6A in females, and we don't see that in males at all. We saw the same thing when we look at EQTL. So again, Alzheimer's on the x-axis. And here, looking at heel bone mineral density, we could stratify that into males and females, and we could see a peak in the females and not in males. So we don't know too much about this gene, but we saw that there is some immune relationship between Alzheimer's and osteoporosis that might be mediated by this MS4A6A gene. Well, the hypothesis came from clinical data alone, then we layered the knowledge network, then the GWAS data. So what are we doing now? We are working, Alice is continuing this work. It's available as a preprint on medRxiv. This is in close collaboration with Pedro Pinheiro-Chagas, who's a faculty member at the Memory and Aging Center. We have access to the clinical notes. We've been mining the clinical notes using large language models to extract specific concepts and integrate them into traditional AI. So it's a combination of generative AI and traditional AI. and see if we can improve our classification. And we do. We see a slightly better, well, a significantly better area under the curve. And we're also able to distinguish not only between Alzheimer's patients and controls, but Alzheimer's patients and frontotemporal dementia. So to summarize this work, we demonstrated that we could predict Alzheimer's, at least with some accuracy, up to 7 years prior to diagnosis using electronic medical records. We saw that some of the clinical predictors were sex-specific, and we dug into the osteoporosis relationship between Alzheimer's and osteoporosis in women. We used the knowledge network approach with Sergio and his team to understand the molecular underpinnings of these relationships. And we layered GWAS data to be able to explore and validate it with another layer of information. Of course, these models need to be validated and hopefully implemented in the electronic medical record. When we think about that, we also need to realize, so what? So what if somebody knows that they're more likely to develop Alzheimer's 7 years from now? Is there an intervention? So that's why we're working on the drug discovery aspect as well as the predictive aspect at the same time, because they go hand in hand. Maybe there are some behavioral interventions that could be done in the meantime while we don't have very good drugs. Or maybe these patients could be candidates for clinical trials earlier on. And of course, we want to explore the biology and therapeutic potential of MS4A6A to understand what might be going on there mechanistically. There's a lot more work that we're doing in my lab looking at social determinants of health and Alzheimer's risk, as well as cardiovascular disease. We've been applying unsupervised machine learning approaches to better understand subtypes of Alzheimer's. And then we've been looking at links between Alzheimer's and specific diseases. So I mentioned autoimmunity as a research area in my lab. Turns out there's a link between autoimmunity and Alzheimer's, also that's sex-specific, that we've been exploring using electronic medical records. And this work came out on Cell Reports Medicine last year. So there's a lot to do. There's a lot of data that is available, both public data as well as clinical data provide an amazing avenue for exploration. And really, we're at a time where we can try and model this biological complexity with AI models, whether we look at DNA, molecular, cellular models, tissue models, physiological or population level models, bringing it all together. And there's a number of faculty here at the Baker Institute working across the spectrum, and I'm hoping that we can maybe use knowledge networks to bring it all together in the future. With that, I want to thank my lab. Ali, Alice, and Yu Chao led the work that you heard about today, but also in collaboration with many, many others. Our collaborators at Mount Sinai early on, collaborators at the Gladstone Institute, Yudong Huang and his team, as well as Dr. Butte, as well as everybody at UCSF for creating the resources that enable many of these studies. These are our funding sources over the years. This is the team, and I would love to take any questions in the time that we have.
Thank you so much, Dr. Sirota. We have 5 minutes left, so we're gonna start taking questions. Kurosh.
Hi, thank you so much for your great presentation today.
Sure.
You kind of mentioned this a bit in terms of the matching and the demographic angle, but I guess with something like Alzheimer's, this disease can continuously progress and it's highly dynamic over time. So I just wanted to know a little bit more in terms of the demographic space, how you guys are kind of accounting for the disease progression in your models. So just a little more info on that would be amazing. Thank you.
Yeah, absolutely. So in terms of demographics, when we look at and develop our case control groups, we match by that. That's our best way to account for demographics, which is not ideal, but we try. There's another study in my lab where we've been looking at social determinants of health, especially in the context of Alzheimer's, cardiovascular risk as a precursor to Alzheimer's and how that might be mediated by social determinants of health. It's sort of a work in progress for now. So we try. The clinical data can or is already matched to social determinants of health information like area deprivation index and so forth. So we're It's hard. I don't have a good answer, but we're trying to model it in different ways. Also, when the models hopefully start getting implemented, we'll be able to see, do they hold up in other places in the US that have different, demographic distributions of patients? And for now, we've been working on the coast, but there's also the rest of the country, which looks quite different. So there's a lot of work to do in that space, but we're actively thinking about it.
Perfect. Thank you. Thanks so much.
Nico?
Oh, thank you so much for the talk, and I learned a lot, and it really got me thinking. My question is sort of similar, but on a different level as Courage. So since you introduced this as a general framework rather than like a AD specific
Yep.
story, so I wanted to ask at the framework level, when applying this framework to other diseases and like other decision domains, like what are the components do you think would largely hold up and are reusable? And what are the ones you would expect to need real adjustment or careful stress testing before proceeding? Because like, are there any like things that are may not necessarily hold outside of AD or across different diseases?
Absolutely. So we are actively applying this framework both in terms of the EHR analysis, haven't gotten into predictive modeling, but more unsupervised clustering, as well as drug discovery for endometriosis, which is also a very complex disease. Well, of course, there are some differences that are obvious. Endometriosis is just in women. So, and the demographics as reproductive health span women. So we're looking at a different kind of different age distribution. So we match by different parameters. For the drugs for every drug, it's a little bit different because we match by the indication of that compound. So depending on what drugs come up, we match by the indication of that compound. So that needs to be adjusted, and it can be automated in the future, but for now, there's some, we work with a clinical expert in the field. I'm not a doctor, so I work with clinical experts in the relevant field to figure out what that framework should look like as we explore different therapeutic candidates. And considerations for Alzheimer's is very, very, like if you are choosing a drug repurposing, I'm jumping around, but there's a part of it that's clinical data analysis where all the things that we match by are different, right? And you can adjust those. But also there is the drug discovery aspect and which drugs you end up choosing. And the considerations for safety are very different, whether you're looking at women who are reproductive age span and might or probably are interested in having kids versus like you would not necessarily consider a cancer drug there because endometriosis is a disease that goes on, like it's a chronic condition. So it's different. Alzheimer's is also chronic, but those individuals will likely not be having children, right? So we think about what are the risk profiles that would be appropriate in that given situation versus not. But you're asking the right question. Like some things can be carried through, like the methodological things. Others are just, it's more complicated and you need to think through them.
Thank you so much. It's very, very interesting and very inspirational and got me into a lot of thinking.
Very good. That's the point.
All right, we are at 2 p.m., so we're going to end the seminar now. But thank you so much again for your great talk and for Q&A session. Really appreciate it.
Of course. Please reach out if you have questions. It's just first name dot last name at UCSF.